Hello everyone, my name is Bhagwan Singh. Today I'm going to be discussing Learning Objective 4.6, Outcome 11, DNA versus RNA, a compare and contrast. What makes them different and how are they similar? Now let's get into it. I'm going to begin by breaking down the differences through highlighting their structure, function, and nitrogenous base. So to begin with structure, I'm going to reference the picture in the top right. One of the key differentiating factors is that 2' spot on the 5-carbon sugar. As you can see, deoxyribose, which makes up DNA, has a lone hydrogen on that 2' location. Whereas ribose sugar that makes up RNA has a hydroxyl group in that 2' area. Keep in mind this is also how they are named: DNA, deoxyribonucleic acid, and RNA for ribonucleic acid. Another thing to look at is the strands of DNA and RNA. A common theme throughout biology is that structure has direct relation to function, which is the case here as well. DNA's double-strand helix is pertinent to its ability to replicate and store information. On the other hand, RNA's single strand allows it to serve as a template for making proteins and then be able to be discarded once its job is done. That brings me to my next point. The double strand makes DNA structure sturdy and stable, protecting the encoded information within. The single strand gives RNA the ability to move and fold in the necessary manner for protein synthesis. Now my final point for this slide is on DNA synthesis in 5' to 3', whereas RNA's is in 3' to 5'. This is because it is more energy efficient to work down rather than upwards, and also allows for the complementary bases of the pairing strand to remain inside. Moving on to function, DNA is often said to be as a blueprint, and rightfully so, because DNA provides key information and conducts growth, development, and reproduction. RNA, on the other hand, plays an integral role in the synthesis of protein. And RNA begins protein synthesis by first getting instructions from DNA, where mRNA is synthesized in the process of transcription. The mRNA is then ready to direct the synthesis of protein through the process of translation. Moving on to our final major difference, I'm going to highlight the nitrogenous base. There is generally overlap over this base, but DNA and RNA have their own specialized bases. So DNA has thymine and RNA has uracil. Further into the presentation, I will cover the rest of the bases. Now I'm going to discuss what makes DNA and RNA similar by highlighting the nucleobases or nitrogenous bases and the sugar phosphate backbone and its role in protein synthesis. So to describe the breakdown of the nucleobases, I've constructed this hierarchical scale. It starts with nucleic acid, meaning DNA or RNA. Then there are two groups of bases divided into purines and pyrimidines. Purines are comprised of adenine and guanine, whereas pyrimidines have cytosine and thymine depending on whether it's DNA or RNA. Next, we have the sugar phosphate backbone, which provides the basic structure for both DNA and RNA. Highlighted in yellow, that'll be the phosphate group that forms on the other side of a strand. Both nucleic acids will have a 5-carbon sugar at the center, where a nucleobase is bonding at 1'. Lastly, I'm going to discuss how important DNA and RNA are to protein synthesis. DNA provides the instructions or blueprint for the protein that needs to be synthesized, and RNA itself is what really conducts the brutal protein synthesis, like I explained earlier. So that concludes my presentation. Thank you for listening. I hope it's informative.